Hi, today we're going to do DNA extraction from strawberries, and those are our strawberries, using some household products. So this is something that you can do easily at home. We are going to use water with salt. I've already put the salt in here. Okay, so this is water with salt solution. Okay, and we are going to actually put in some soap, some dish soap, because it has detergent properties which will break the cell wall open from the strawberry cells to get the DNA out. And we need a beaker here, and then I'm going to be using isopropyl alcohol. I've seen it done with isopropyl alcohol, or you can use ethanol.
Or rubbing alcohol.
Yeah, that's rubbing alcohol. Isopropyl. This is my Kayla. And we have our strawberries. We took the tops off of our strawberries, as you can see. And we have a pipette here.
Which is optional.
That's my other, Miss Daniel. We have a pipette here for scooping out our DNA. Okay, so we've already added our salt to our extraction solution. Now we're going to add soap to it.
Is it a tablespoon?
It's about a tablespoon of soap should be sufficient.
Can I mix it?
Sure.
Can I?
Okay. All right. And this is my Sophia. She wants to be part also. And while they're mixing that, we're going to have someone smash the strawberries. Let's have Sophia. Sophia, you start smashing. Okay. So we'll talk a little bit about DNA first. DNA is found in all living things.
Smash it.
DNA is found in all living things. Anything that has cells in it will have DNA in it, and DNA is found inside the nucleus of the cells. Okay, and you can smash some more. We've done the smashing. And DNA is a double helix, which means it has two equal strands, and it has base pairs that link together. So the A's will always go with the T's, and the G's will always go with the C's. Okay, so that's how the DNA, the strands go together, because the A's are attracted. The A on one strand is attracted to the T on the other strand. The G on one strand is attracted to the to the C on the other strand. And so when you get all of these strands together, all of these base pairs coming together, they will attract one another, and they'll make a double helix. Okay, and so that's what you have. And let me show you a picture of what that DNA double helix actually looks like. That's what a DNA double helix looks like. Okay, so what you would have is the A on this strand attracted to the T on this strand. Okay, and then they form a bond here in between. Okay, and so everything that's living would have DNA in it, and everything that is living or was living at some point will have DNA in it. All right, because DNA replication is important for making new life. Okay, so now we have our strawberry mush here.
It needs to be squished because there can be no more chunks.
That's fine. We have our strawberry mush here. Okay, and I'm gonna get one of the other ones the little ones to hold the camera for me. Could you hold the camera on with our experiment? Okay, and so what we'll do is we'll take our DNA mush, and we'll use a strainer here. I'm going to let Sophia do it. Come on, Sophia. You can pour that into the strainer. Go ahead, quickly. Okay.
Slowly.
not so slowly with this. There are some things in this experiment that have to be slowly, but not so much with this. Okay, and
You have to
can
use the spoon to mix it
spoon
around.
like this to just strain out all the juice. Want to do that? Go ahead. Very good.
Mom, can you move your hand?
Very good, Sophie. Very good. Okay. So we mash it up nice and good.
Sometimes it takes too long to get in
It's well mashed.
or to get out.
Okay. And we don't want to disrupt it at this point. We want to allow the extraction mixture to work. And what the extraction mixture is doing at this point, not yet. What the extraction mixture is doing at this point is it's breaking the cell wall. Okay? It's breaking the cell wall of the strawberry cells. Okay? And we are going to use alcohol now.
Tip it over.
We want to use alcohol now, very gently. Has to be poured in very slowly and very gently. And that is what's going to bring our DNA out of the solution. Okay.
Whoa. 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 Hey guys.
We'll leave that alone for a few seconds. And we will be able to clearly see the DNA that is extracted.